Hello! In today's lab, we are going to be exploring the human origin made possible by PCR amplification of mitochondrial DNA. Before we do that, we must first understand just what mitochondria is and all the things that we can understand from it. Mitochondria is found in most animal and plant cells, and it is a double-membraned organelle. As you can see, this is the outer membrane, and then here's the inner membrane, and it is accompanied by those little folds. Those are called cristae. And mitochondria is believed to have originated by endosymbiosis, which happens when a single-celled organism engulfs another one, and then they work together to make one beneficiary organism, which is mitochondria. The breakdown of mtDNA can help us understand many things, including how we survived through the Ice Age, how past societies made it through, why we're susceptible to certain diseases, and things like that. We are going to be using mtDNA because it replicates much faster than nuclear DNA does, and it will make for a less time-consuming lab. The mtDNA also contains the genes that are essential for the proteins in the electron transport chain. It produces ATP, which is adenotriphosphate, and also has mitochondrial-specific RNA. And the mitochondria is a reflection of the maternal lineage, as you do not receive any mitochondria aspects from your father because no mitochondria is passed down through the sperm. So next we're going to be discussing what PCR is and all the cycles you have to go through to produce a new DNA sequence. So using heat-tolerant bacteria called Thermus aquaticus, which we will call TAC for short, DNA polymerase is extracted from TAC and then combined with a minuscule amount of DNA segments because PCR is a very sensitive process, and you can't put in big chunks or it'll just make it go all out of whack. And then we're going to combine those DNA sequences with a buffer. I believe we are going to be using lysis buffer and primers to undergo 3 different cycles to produce bigger sequences that makes it easy for us to analyze. So the first cycle is called denaturation, and that happens by using high temperatures to denature the proteins, which breaks the hydrogen bonds in DNA to break up the DNA sequences. And then the second cycle is called annealing, and that is where we rapidly cool the mixture, and it makes it possible for the primers to base pair with DNA sequences. And then the last cycle is called extension, and that is whenever we once again bump up the temperature to the optimal temperature that TAC polymerase likes to work at. I believe it is 72 degrees Celsius, and the TAC polymerase will add nucleotides to the primers to synthesize the new strands of complementary DNA that we will be analyzing. And fun fact, each cycle of PCR doubles the amount of DNA material that we have in each cycle. So today's lab, we will be extracting cheek cells using a swab, and similar to last week's lab, we'll be isolating the DNA, and then we will use PCR, the PCR machine, to generate bigger sequences of the DNA, and then we will inject that into agarose gel and analyze it through electrophoresis. So first, we will take 10 milliliters of saline solution, swish it around in our mouth, spit it back into the cup, and take out 1.5 milliliters of the cheek saline solution and send it through the centrifuge. And after it comes out of the centrifuge, we make sure that all of the different elements are in their own places and there's no clumps of cells left. We will place the centrifuge mixture into 150 microliters of lysis buffer. And after giving it a good shake, we will cap the tube and place it in a 55 degrees Celsius water bath. And after 5 minutes of being in the water bath, we will take it out and shake up the tube and incubate it in a 99 degrees Celsius water bath. After 5 minutes of being in the 99 degrees Celsius water bath, we will centrifuge the tube for full speed at 2 minutes. And now it is ready to have 50 microliters of it transferred into a new, fresh microcentrifuge tube. And then after we put it into that new microcentrifuge tube, we will then place it into a freezer or ice bath until we are ready to conduct the PCR. So now, we're going to be sending it through the PCR. And to do that, we must obtain the tube of DNA that we just freshly prepared. And before we get started, we should label it with our initials so that it doesn't get mixed up. And we will add 20 microliters of the mitochondrial primer mix, right here. 5 microliters of it are extracted DNA, and a PCR Evo bead plus. PCR Edvo bead plus. Then we will give it a slight shake and gently mix it all together until that little bead is completely dissolved. The solution should be light orange or amber in color. And now we will place this sample, after everything's mixed together, we will place it into the centrifuge machine and take out the sample that collects at the bottom of the tube. It should be like a little clump of cells again, probably similar to the isolated DNA that we had in last experiment that kind of looked like gelatinous. And then we will place the tubes into the PCR machine, which is right here, and it will undergo the 3 different cycles of PCR that we previously mentioned. After that, Dr. Wang will hopefully already have the agar gel prepared, and we will take out small samples of our freshly PCR'd tube and place it into the little designated slots, and we will be able to analyze it by seeing the different fragments and how they move through the gel, made possible by electrophoresis. And this is just a picture from last week. And Dr. Wang, if you have any questions, please feel free to email me. Thank you.